different depths is that there is sort of
we'll find out in a second.
zonation. It's not obvious zonation, but there's some zonation by depth in the water column. So we will see animals down here at 800 meters that we will not see when we do shallower transects. And that's because the pressure, but more importantly, the temperature, and generally even more importantly than that is the available oxygen in the water column at any given depth is one of the determinants of what, or sorry, some of the determinants of what animals you see throughout the water column.
Let's get the jellyfish here. Let's start the transect.
Okay.
All right, watch lead. This is your ROV navigator. I'll talk on the OKX2. This marks the beginning of the 800-meter transect, 2003 UTC.
All right, Andy, if you keep an eye on my...
So right now we're looking at a large jelly medusa called Solmissus. It's a pretty common predatory medusa in the midwater. And actually it can get the size of, say, a dinner plate, which is pretty uncommon for animals that we see in the midwater. Like I said, this is a predatory species, and one of the things that it commonly eats is other gelatinous organisms. Okay. So one of the things you'll see during these transects is gelatinous organisms are very common.
You just gotta let me know what's going on.
So of course,
I will tilt down now.
There's a lot of common animals, then there's going to be predators of what's available.
And slightly down, you're at 803 now.
We saw a fish going by the upper camera.
What's that? I'm not talking about the signs.
I didn't get enough of a look to see what kind of fish that was.
I was on OKX for the announcement of the beginning. Wrong.
There are a few very common small types of fish that we encounter in the water column. One of the groups are myctophids, and actually we just saw that small black fish that shimmered and went off to the right. They have really large, very reflective scales that actually fall off very easily. That's one defense that they have against predation, is if something grabs them, they can actually drop some of those scales and potentially get away. Right now we're looking at a little,
come on out.
Looks like maybe a mysid.
803.
Or krill, perhaps.
70. Copy that.
Yeah, myctophids are very common small fish in the water column.
Shiny top left.
So we'll probably see quite a few of those.
About right.
It's a little fast.
See the shiny on the left edge?
I don't. You have to.
We can slow him down by moving the ship backwards.
Yeah, that's what I'm thinking.
If he wants, but I don't.
Might be too much though.
Might be too much.
You know what I mean?
I would move the ship back at like 0.1.
Okay.
It's coming by
reverse of your heading.
To like.
What'd you say?
210, 220, something like that.
Oh, what are you asking us?
He's gonna move the ship back at.
Yeah, 225. Right. 225.
Okay. Just math.
Just math. Okay, I'm gonna follow you up a little bit. And I am now at 45.
So hey, Bridge. This is ROV Nav.
Small jelly over on the right.
ROV Nav Bridge. Go ahead.
Yeah, we'd like a ship move, 50 meters, bearing 225 degrees, speed .1.
Good copy. That is 50
Actually, on the bottom right, you might have seen a small, long, narrow animal. I think that one might have been a Keatingnath.
It's a good copy, Bridge.
Here we've got a fish right in front. That actually is the other very common type of fish we see in the water column. The genus is Cyclothone, and actually they are. I've heard they are the most common vertebrate on the planet.
Let's get this guy right here.
So they're pretty important. They're small, as you saw, but again, they're few and far between. But when you add up how many there are in the water column versus the volume of the whole ocean, there's quite a few of them.
Ships should start moving here shortly.
ROV Nav Bridge, move initiated.
Move initiated. Thank you, Bridge.
Okay, I'm going to tilt down. Yeah, I need to get
the center right here.
You're hearing the navigator speak with the bridge a bit, still kind of adjusting the ship with respect to the current so that we can get a nice amount of animals moving past the camera for us to see what's occurring here at 800 meters.
Come in.
So we've got another small fish here. That looks like another cyclothone. They have sort of a pointy snout, and they're often pale like that. And if you get a really close look, they look a little
tilting down,
Skeletor-ish to me. So if you can get a good look at them, which is difficult, they move quickly sometimes. It's pretty easy to tell between cyclothone and Myctophid.
Larvation housing.
Yeah.
Looks like a radiolarian right here. So radiolarians, single-celled organisms. Just find them in the water column, and they actually collect some of this marine snow. So all these other small particles we see going by. There's actually a lot of unused nutrients that some animals down here take advantage of.
Let's get this guy as we come by. Let's go ahead in.
You're starting to turn pretty far, of course.
Yep. I'm lateraling here.
Okay.
Hey, come on out. How much time we got here?
3.5 minutes.
Is similar to how Medusa swims, where they move those lobes outward and then pull them together quickly, and that acts as a jet where they displace some water and then moves them backwards.
Passing 9-0.
Looks like we've got another.
Let's do a snap real quick.
Ctenophore here. Yeah, that one. It's got this very bright white spot in it. That is the gut, and it looks like it has eaten semi-recently a crustacean, so possibly a krill wrapped or curled up inside the stomach of that ctenophore.
Moving a little fast here, Stephanie. I gotta push out ahead a little bit.
No worries, thanks. Another few cyclothone on the right and down below that we're passing by.
Passing 6-5 degrees.
Right.
Looks like we're coming up on the right side. Another, the common name is the dinner plate jelly. I mentioned that they can get the size of a dinner plate, hence the common name. So this, but the Latin name is Solmicis, and one of the unique things about this jelly and the group that it's in, they can actually trail their tentacles behind them, like we saw on this particular individual, but they can also
it hasn't hit you guys yet.
Bend the tentacles forward, and so as a predatory jelly, they actually lead with those tentacles, and the tentacles are covered in stinging cells. And so if you're moving forward with your predatory apparatus, it's probably a pretty good strategy to encounter a meal.
Coming in, Roland?
This looks like it might be the remains. Oh, actually, no, it's still inside. So this is a larvacean, and they look a little bit like a tadpole, actually. So if you can see it beating its tail in the center of this web. This is another type of animal in the midwater that that secretes a mucus web and actually filters out all of these particles that we see all around in the water, again, the marine snow, and they actually feed off some of those particles, but they have, we could see very clearly the outer house. They also have an inner house, which is very complex, and they actually, with different kind of sub-chambers within their filter feeding house, and they actually can sort out the particles by size. So Again, they have 2 different filter houses. They have an outer house that kind of gets out the large particles, and they shed those every once in a while. And the inner house can sort by size. This looks like it could be a larval cereanthid anemone. So actually, if you were tuned in earlier today, or even yesterday, we saw some good-sized tube-dwelling anemones on the seafloor.
Get the low one.
That may have been the larval stage of one of those.
Wings are all the way upward.
And right now we're looking at a salp chain. And again, you're gonna hear me say this over and over again. So many of the midwater animals are feeding on the particles in the water column across multiple different phyla.
What lights?
So
the front lights.
Salps. This is a chain, so there's you can see each of those opaque structures is a stomach of one individual in this colony. But each individual is sort of shaped like a barrel or a tube. And they open and close their body or their tube and swim through the water, filtering particles, and water comes in one end, and then water and fewer particles go out the other. And again, they're using mucus on the inside of that barrel-shaped body.
This is our last subject. After this, we need to start coming up.
Capture their capture food. So
this is the end of the 800-meter transect.
Okay.
We're going to start coming up after this watch leads.
Okay, yeah. So that was the 10 minutes. Actually, we saw lots of great animals in there. We're going to come up to 700, I believe.
So we need to turn the mains off.
Yeah, mains off. 10 meters a minute. Normal
Navigator,
config.
this is watch 2.
Camera platform at 45 degrees. Rotator. D2 is at plus 42. Looking past the bars. Mains off.
Okay, and I got to come up to 45. That's 45 for me.
Plus 42, you said?
Yeah.
That's really high.
Yep.
Right. Oh, yeah, and they
plus
zoom
42.
in.
Okay. Zoom in past the...
All right, rolling.
All right.
You're coming up a little bit already, so I'll start coming up.
So we're moving the ROV to a shallower depth to do another transect.
You just stay in the middle.
We'll probably need to readjust the ship because even a difference of 100 meters in the water column can have a drastically different current regime. Could be coming from the totally opposite direction. So we'll probably have to reassess as we set up for the next transect.
Next stop, 700 meters. Pilots.
Copy.
We're going at 10 meters per minute.
So Heather, the Okeanos Explorer hosted a Reddit AMA yesterday, and we got lots of different questions. And I was wondering, since while we're transiting to the next location, do you want to answer one of them live? I think the main one people kept asking was the coolest thing we've seen when we've explored the water column.
What's that flare?
Sure. So are we talking the coolest thing we've seen by using ROVs, or some of the other work that I do, for example, with networks?
Oh, no, I'm talking about PTZ.
Whichever you think is cooler.
The other lights are pointed in.
Well, so I think each tool is a great addition to doing deep water science. So when we do these ROV transects in the midwater, it's super interesting because we get to see behavior, how they actually work in situ, and what they're actually doing in the water column themselves. The hard thing with ROV work, and especially for cephalopods, because they're pretty quick, so squid and octopus move pretty fast, so oftentimes they're not in the camera shot. The gelatinous folks love the ROVs. It's great for them because the jellyfish and the ctenophores and other organisms like that are pretty slow-moving and might not react so quickly to a big ROV with lights and sound coming at it. But we do get some great cephalopod footage with that as well. So I think some of the squid footage I have seen from past cruises we've done with Okeanos has been really enlightening to give us a little bit of insight about how they act and what they do. And with the net system, so I've been fortunate enough to go out to sea and do some collecting with very large trawl nets where we collect big adult organisms.
Which I guess
and then we've used some other net systems where we catch juveniles and smaller organisms. So it's, I think every tool that I use always has a pretty cool factor to it. So I would say some of the squid footage from ROVs, and one of the most fun ones that we did, there's a lot from the net collections, is for my PhD work, I was working on squids and octopus from around Gulf of Mexico, Caribbean, and a little piece of the Atlantic around Florida. And we didn't know how many species were there for cephalopods. And we had seen giant squid, things like that we've documented. But there was another large squid that was found, fishermen. They thought it was a giant squid, and then they called a colleague of mine and we're like, hey, can you come identify this? We think it's a giant squid.
Move forward or
and we get to it, we're examining it, and it's not a giant squid. It was large. It was a 20-foot squid from the end of the tentacle to the top of its body. But it was not a giant squid. It didn't have any of those characteristics. So we did a lot of research and come to find out that it matched very, very closely by morphology to a squid that was found off the coast of Japan. So we were fascinated because we're in the Gulf of Mexico finding this. It matches a creature from off the Japan coast, and it matched morphologically. The one piece I wish I would have had from that 1960 specimen off Japan was DNA, because then we could have matched it genetically. So I ended up using part of that in one of my PhD chapters, and it was this new squid called Asperoteuthis acanthoderma to the area of the Gulf of Mexico. So, and that was in 2007 and 8 where we found a couple of these specimens. And then the fun thing for me since has been that all these cruises I've been able to go on, we keep finding Asperoteuthis acanthoderma. So now I've got a male, a female, juveniles. So now we're getting the whole life history of this creature that I think has been there for a really long time, and we just haven't been able to collect it. So I think there's a great value to all the tools we use for marine science, especially when we're looking at open water exploration.
That's great, Heather. Actually, you were talking about behavior and being able to observe animal behavior with the ROVs. Do you can you touch on how you think that Asperoteuthis is feeding, given the unique morphology of their tentacle clubs, and maybe talk a little bit about that as well compared to other squids?
Well, we really don't know. We actually did get some footage, I believe it was on one of the Okeanos cruises, of an Asperoteuthis in the Gulf of Mexico, I believe. It was either last year or the year before, maybe a couple years ago now. But the tentacles have this large photophore at the end of it. And some of the other squid do too. These guys aren't the only ones with photophores on their tentacles, but it could be thought that they use this to lure over other organisms. And as to what they eat exactly, I don't know. We know in general, squid eat things their size. So these 20-foot Asperoteuthis squid are most likely eating fishes, crustaceans, other things that are about the size. They have to chew up things really, really well with their mouth, so they can digest it properly. So we think they use their tentacles to grab for prey and then bring them up to their mouth, where they kind of use their arms and everything to protect their mouth region while they break down their food.
Great. Thanks, Heather.
Sure.
Yeah, so now I'm imagining, what kind of fish they're going to be catching out there that are near to or similar size to themselves.
Yeah, I think they'll eat smaller stuff too. Like myctophids are like food for everybody, right? So
yeah,
I would imagine they're eating small organisms that they come across. I don't think it has to be a 20-foot fish by any means that they're going to be eating.
Right.
They've got to still be able to get it in their mouth, and chew it up pretty well.
And we've observed, so Asperoteuthis is part of a family of squid, the Chiroteuthidea. And we've seen other members of that family, much smaller members of that family, eating fish commonly. But in other squid families, we see them eating individuals of the same species. So squid can be cannibalistic, I guess. They're opportunistic, certainly, especially, in water column like this. You're gonna want to grab and try to eat anything that you can get your arms around in the case of squid. So they'll take other squid, or they'll take fish, shrimp, and things like that.
Do you think you're going too fast, too slow, Josh? Or about right?
Vertical is fine. I'm just getting pushed all over the place.
Ooh.
And Adrian, you're on the line, correct?
Yes, I'm here. And we also have Amanda now from the Silver Spring APC.
Move complete.
Awesome. Hi, Amanda.
Wait, the move is... Are we going to keep moving?
Sorry, I wasn't here for the start of the dive.
Currents are going to change every...
No worries.
I could keep moving if you wanted, but... How do you guys feel?
Adrian, I was going to ask you to talk a little bit about, since we were just talking a little bit about what squid eat,
stop, I think it's gonna
maybe you could talk about
real quick.
what eats the squid down here.
Well, because the move was only
so
0.1.
I actually, for my PhD, I studied marine mammal foraging behavior, so marine mammal feeding behavior. And I particularly was interested in deep diving tooth whales, or odontocetes. And those whales eat what you guys are talking about, the squid, which in turn eat many of the smaller things like metophids. And we were interested in seeing if these smaller things are attracting your larger squid, which in turn are attracting these large whales. So if you look in, I think it's feed 3, we have the acoustics, the EK60s up there, and right now there's 1, 2, 3, 4 frequencies. And you can see these deep scattering layers that form layers around 300 to about 800 meters, and that's where the small critters are. So I was interested in looking at if these small critters are attracting your squid, which are attracting those large whales.
Cool. And why are there layers of these animals in the water column?
Yeah, so Heather talked a little bit about zonation earlier. And so how you guys heard, we're doing transects within these different zones in the water column. And these animals make layers in the water column based a lot of times on light. So they're down at depth during the daytime to avoid predation. And then at night, they make the largest migration, or the biggest migration to the surface to feed. So they actually stay down at depth around this depth area to avoid predators.
What that looks like?
Yeah, so one thing that to think about with the water column, which it was hopefully sort of obvious, but you may also not stop to think about, is there's no physical thing to hide behind or hide underneath or anything like that. So your greatest protection can be the fact that there's very little to no light depending on what depth you're at.
Back,
but the problem with that is the deeper you go, the less light there is. However, the vast majority of the food is produced and occurs up where the light is.
What'd you say?
So that's why, as Adrian was talking about, there's these mass migrations on a daily basis. At night, these animals swim up closer to the surface to feed.
Between us.
And then they go down where it's darker, but there's no food during the day to hide from visually oriented predators.
Maybe even a little more south. So go ahead and
so we're at 700 meters now, and so we're setting up for the next transect.
Go 0.2, and then we'll dial it in from there as he goes along.
Bridge, this is ROV Nav.
I'm going to let you push out.
Yeah, I'd like to move 100 meters, bearing 190 degrees.
Coming down.
Speed decimal 2 knots.
Yeah.
Good copy. All right, guys, let's get going.
Okay.
Okay, I'm looking down on you.
I see in the
9-0.
upper left there's maybe a dragonfish, a really long, skinny. And up ahead we have a type of ctenophore that is called
kick those extra
either
lights on.
cestum
Way
or
below me,
velamin.
Roland.
Yeah, that's-
You can turn them on.
Actually,
Those lights are
usually
actually
I
aimed
think of
down.
these as occurring
yeah, this
shallower
is,
in the
this
water
is
column,
literally,
but
like, below me.
then again,
There you go.
I have done most
Okay.
of
Okay, you gotta
my
push out.
midwater research in the
Kill the
Pacific,
lights.
so I'm unfamiliar with the Atlantic water column. Other than some work
Move
I did with
initiated. Thanks,
Amanda.
Bridge.
Pushing ahead.
Okay, I'm all the way back.
Are we still getting set up here?
Yeah, we haven't started quite yet.
I'm
following
taking over the scribe annotations here,
Got you at 9-0.
but we can't. We don't have the pilot swooped in right now. So if you can
Oh, okay.
announce it when we start
Yeah.
the next transect. And
Yeah, I think the
that'd be-
navigator
getting
announced
that
it
taken.
on the OKEX channel earlier, but I'll repeat it.
Okay, thanks.
I'll let you know.
All right, now we're
Ready-
starting to look good.
You ready
Yep.
to start
Thank
it?
you.
Yes.
All right.
All right, watch lead. This marks the beginning. I'll talk on that channel too, if you'd like. This marks the beginning of the 700 meter depth transect.
Awesome.
2028.
We did hear that.
Cool, yeah.
I'll get the arrow
Thank
along
you,
at the top.
navigator.
Coming down with
So
you with
about
camera.
approximately dead center, now going off to the left, I see a krill.
Gotcha,
At least I
6-0.
think it's a krill. Again, that's a
Good
favorite
display.
food of some of the ctenophores and cnidarian medusae that we see in the water column. And this is the remains possibly of a larvacean house. So in the last transect, I was talking about how they
Got you
filter
at
out the particles using
8-0.
mucus houses, and they actually,
Pushed sideways.
when the
too.
outer house gets clogged with particulates, they abandon that
I
house,
think if we,
and then it becomes what we call a sinker.
turned maybe
And
10
so it's
degrees or
mucus
so.
with
Maybe even
particles
more than that, but
entrapped in it, and
Like,
just the fact that it's all conglomerated causes it to sink relatively much faster than any of those particles by themselves. So it's a sort of a more rapid transportation of nutritious material to deeper layers, and possibly also all the way to the seafloor.
pushing ahead.
I think stuff's still coming at us too fast.
Yeah.
So
it's going
siphon
to take a
for
moment for the
Come on
wire
in.
to get down there.
Going below me. Let go.
So siphonophores are colonial organisms.
What do you think now?
The one end
I can
is
increase
a
0.3.
float
Well,
and
think
some
it's worth it.
swimming bells.
Should we wait
Serial
That's
started moving at all?
It's
Gets
hard to
the
say.
Can't tell.
siphonophore to move around, and
I could
the
try
sort
to
of the
judge by
tail
high
end
pack,
We
of
need
the
to keep
siphonophore
serials.
is a
If
bunch
the
of
ship is moving, we need to keep that.
zooids that
going.
are essentially stomachs
Starting and
and
stopping.
reproductive material,
It's not- I don't think
and
it's gonna
they
work.
have stinging tentacles
And
that
then
branch out
as
from
we
there.
get to a depth,
That's how they
we'll adjust the ship move
get their next
to
meal.
whatever we're seeing.
So fish
That way we're at
and
we're at a
squid
constant rate as
and
opposed to
any
stopping. and
basically any
swinging
swimming
and
animal can get
all kinds of
entrapped
stuff
in
going
those
There'll be a
stinging
little vector
tentacles
map,
if
but
they
that'll be
swim.
easier than
If they're
this.
swimming in the path of where those tentacles are splayed out
get
as
this guy
a
up
sort of
here.
feeding net in the water column.
You have a few seconds. I'm looking back at you.
Not actually sure. Does anybody have a guess what were those
let go.
paired
Please,
things?
those were phylodendrid bidarians, which are a single-celled organism
What if we just do a drift
that we
test
do
next
sometimes
time?
see kind of clustered
Cluster.
together.
Fine.
Oh, okay.
Stuff is flying by
replicated.
serious.
They're in nice places.
You can try 0.3.
Wow.
We saw a lot of those out in the Pacific. As you were saying, I'm also really new to the Atlantic. So
We got a heck
that's
turn
what
already
it
up,
looks
though.
like to me.
Nice. Yeah, I feel like I've seen them a couple times, but I
speed to decimal 3.
someone probably told me what they were,
Thanks.
but I didn't
Following you
retain
up.
it. This actually looks like
Looking down to right now, 9-0.
a paralipidid. Would you agree, Amanda? That's
I do agree.
so very long, skinny fish that we often find vertically oriented in the water column, and then you can see he's moving at a fairly decent clip, but you can see that it's just the very, very end of the body that appears to be,
Pushing ahead.
moving it. Yeah, we're doing 10-minute or so midwater transects right now. Earlier we did one at 800 meters. Right now we're doing one. Actually, we're at about 680 meters.
The ADCP is not ready.
Oh, it's physically off. Okay.
Yeah. No ADCP.
And we're moving along at a set depth,
It's
or
not
with it,
working.
plus or minus 5 or 10
Well,
meters.
that's clearly not working, but I'm
That's
talking about on
see
the
what
ADCP. It's
midwater
now on
animals
its
that we
computer it's
encounter.
working.
Okay.
Shiny fish top center.
It was 10 minutes ago,
Only the shallow one.
or an hour ago.
Andy.
Yeah, still goes down to about 1,000 meters.
It's a small fish there.
The one that
That's
we have.
maybe Amanda. Can you
What else?
identify that type of fish?
It was hard for me to tell from here without zooming in.
Yeah, it's moving pretty quick and erratically.
Yeah. Be reflective, but I think so much more than that.
The percent good is not good, Andy. I have
What's the other little tab?
2 whiskers. That's off.
Okay.
Sorry.
Oh, there it is right there.
Here we go. Okay.
Object below.
Starting at
Go ahead, Ed.
600 meters, we got
Mucus.
Right now we're looking at
Southwest.
that. Looks like it possibly part of a molt
At a
from a
magnitude of-
suggested shrimp. Either that or a mucus string
0.25.
remains from some feeding structure.
Bright object.
And here
of
we have
pink.
another type of siphonophore. So you can see up at the top right,
So what'd you end up with?
Go ahead, Ed.
it's the swimming
0.25.
bells. You can see them
0.25.
propelling this
Meters per second.
animal
Hold on. I'm gonna
upward and now sideways.
That's like
All the
0.5
pink
knots,
structures
0.5
are
knots.
the
Southwest.
Coming down.
retracted, stinging tentacles
0.5
used
knots
in feeding.
southwest. Oof. Okay. I need a bump to 0.4. Got you all the way back, 101.
You want me to increase that to 0.4?
We're flying right now. I mean,
Yeah.
I would give it a try.
Bridge is RV now. Yeah, could you try increasing that move to 0.4, but if that's too fast for you, you can go back to 0.3.
So I don't know if you
Great,
guys
thanks.
heard
I
that,
want
but
to think about
navigators
nav,
communicating with the bridge to
our angles as well.
adjust
we
the
had to
speed
turn
of the ship,
substantially.
um,
250 degrees.
probably because of changing currents, things like
I
that.
figure it might change a little bit, but mostly it's going to be southwest. So as long as
Okay.
I'm moving out southwest, I think that's going to be.
Let's get this
it's
guy
gonna be
real
what the team
quick.
needs. But I'm going to have to stop the ship to change that angle. So and Josh said he does not want to stop it. So
You can see a lot of the animals in the water column are transparent. That's one way that
we
many
got 2
different
minutes left,
animals
guys. A minute and a half left.
hide from
this
potential
depth.
predators. Also makes it pretty difficult for
You shouldn't have
us
to stop the
to
ship to change
focus the cameras on them
angle
when
of
they're transparent.
motion.
Yeah. We're doing ship moves. Overground. DP.
Yeah.
I can ask for him. Bridge, this is RV Nav. Yeah, I have a question for you. If we wanted to change the angle of the move, the bearing, do you have to stop and then type a different move in, or can you change it on the fly?
One up, okay.
Okay, I thought so. Thanks.
Looks like there's a low bait Tina-4 that just went off screen below right.
Got 30 seconds left at this depth.
Almost done with this transect at this depth.
Let's get this guy.
Here we have a krill. Very abundant in the water column, as many of you probably know. One of the favorite food of some types of whales in some locations, but not only are they fed upon by whales and larger animals like that, but many of the Tina-4s and Medusae feed on them as well.
Okay, let's get this guy. Go ahead, Ed.
This is a suggested shrimp. And they have, as you can see, very long antennae, and that allows them to extend the reach of their ability to sense movement in the water column, whether that be, I guess, another individual of their species, or more likely they could sense potential predators, and they can quickly flick their tails and jet backwards as an escape.
Okay. So you guys got to start
I
coming
know this
up
because we've
to 600.
tried to collect them many times, and they're
Mains
very
off.
difficult to collect because they're erratic and
I'll
quick.
follow you up to 45 degrees.
So that was the end of our 700 meter transect. Now we're going to move up to 600 meters, I believe.
Yes, that's correct. 600. Up to 600 we go. So yeah, Josh, I got this ship moving 0.4 now.
Okay. So you have the.
Go ahead, Bridge.
Got you at 4.5 pilot.
Okay.
Okay, can we tack on another
Seems like
100
we
meters
saw
to that bridge?
Quite a few more sort of larger organisms at the
Ready to go?
800 meter
Ready to go?
transect
Yep.
versus
Heading up.
the 700 meter transect.
Correct.
And that's not
That's
uncommon, given that, you know, we could be in a
Okay, we're
different patch of
give me
water.
a
The
minute.
current is going different ways.
So you have an
and
estimate on
animals
what the
in the water
currents are going
column
to be at
can
the next
be
step?
patchy, so
we
you
can
can go
get the ship moving in the
for a
right
while
direction
and
before we
see
get there. And
nothing much,
see what we have.
and then
Yeah, that's
you'll hit a patch of water where there's just quite a few of either
So
one
you're gonna
type of
figure out
organism
how to get
or
0.1 out
quite
of
a
those
few
currents.
different types of organisms. So you never know what you're going to find.
With the ship move.
Yeah, that's easy.
I think that's a small
Thanks,
fish
Bridge.
out there that just swam away.
Got 10 minutes, we can do it. Bridge is RV nav. I'd like you to change the bearing of that move, so you might have to
Okay,
come
so
to
I
a
just
stop.
realized I didn't tell everyone
quick,
out
and
there
then, initiate
who
the
it is
next
that's
one.
talking to you from the
so
ship.
we want to do 100
And
meters,
So this is Stephanie
and
Bush.
then the new bearing
I
would be
work at the Smithsonian
225
Institution,
degrees, and then I also
National
still want to do
Museum
it at
of
0.4.
Natural History currently, but I've done quite a bit of research in the past on the water column, particularly deep sea squid behaviors, as well
Thank you, Rich.
as pteropod population connectivity. So looking at
All right, pilots, that's going to be
how much or how little
southwest,
movement
which is what my
they
ACP says.
they encounter by riding the
It says
currents around
0.5, I'm doing
to different
0.4.
geographical locations.
All right.
So,
Should be
given
pretty
that,
close.
it'd be great to have the other
Yeah, that's the
midwater
direction, yeah.
biologists and any other scientists that are on
So
the
you guys
line
are-
introduce
you guys are already
themselves,
at, so.
tell us where you work and what kind of research you do.
Good go ahead here.
Initiate. Thanks, Rich.
This is Amanda Nepper. I work here at Ocean Exploration and Research. I'm an oceanographer with the program. Been here for a couple of years, but my background is in midwater fish primarily. But I've been participating in a lot of the Okeanos midwater work, so slowly learning some of the other tax set as well. We actually have a summer intern with us today, so I'm going to go ahead and let her introduce herself.
So this is Gina Seelig. I'm an educational partnership program undergraduate scholar studying marine science at the University of Hawaii at Hilo. And through the EPP program, I've been able to intern with OER this summer. And for the past couple of weeks, I've been helping Amanda, who is one of my mentors, study the abundance and distribution of Pelagosaurus, a pelagic sea cucumber, by going through the video from the Mountains in the Deep expedition on features.
Oh, thanks, Sarah.
Nice.
I'm also with NOAA Office of Ocean Exploration and Research, and my background is in studying whale behavior using active and passive acoustics. But I've also had the opportunity to trawl with some of these deep sea organisms.
This is Heather Judkins. I am with the University of South Florida, St. Petersburg. I work with cephalopods and working with ecology, phylogenetics, lots of
yeah, move's looking great.
stuff.
We do kind of want to almost continuously move at this, so if you can make that range on that move like 300 or something, or
anybody
let
else
me know
on
when
the
you get
science
close to
line?
the end of it. Thank you.
Great. Well, thank you all for introducing yourself and joining us. Also joining us from the chat room, we have Andrea Cotrini from Harvey Mudd College, Tammy Frank, George Matsumoto from my previous research institute in Bari, Lauren Walling from Scott France, and Upsana Ganguly. Sorry if I didn't pronounce your names correctly, but from the University of Louisiana Lafayette. So yeah, you know what our different strengths are. You know, not all of the things out here are my strengths, so feel free to input what we're seeing, or either verbally or in the chat room. So we're just heading up from our 700-meter transect to our 600-meter transect. We're about halfway there. We're at 649 right now. And then we'll be doing a 10-minute horizontal transect at 600 meters to see what kind of organisms are inhabiting that depth.
So what are all these particles that we're looking at, Stephanie?
So we call this marine snow, actually. And the marine snow is comprised of parts of dead animals, whole dead animals, as well as fecal material from much of which of all those things are produced at or near the surface, and then it slowly sinks down through the water column. Very, very slowly, in fact. So yes, as we're traveling around either horizontally or vertically in the water column, this is the vast majority of what we're going to see. But there are many, many organisms that actually take advantage of all this material, because there are still a lot of available nutrients within some of this marine snow. So animals collect via filtering or mucus webs, collect some of this material, and then use that as food, reprocess it. So it does look a little bit like we're the opening scene of Star Wars.
In an ocean far, far away.
Yes. We are...
Currently, right now.
We are, 300 kilometers off the coast now? Something like that?
So we are about 200 miles pretty much due west off of North Carolina.
Okay.
So we're just at the edge of the U.S. EEZ that surrounds the U.S. continental margin.
Thank you, Casey. The reason that we're so far offshore is along the east coast of the U.S., we have to go pretty far offshore in order to reach waters that are deep enough to get down. So earlier today we were doing a dive on the sea floor at about 3,300 meters. So we were traversing across some soft sediment, found some different rock outcroppings. Now we're about 630 meters now. So we're looking at all the midwater organisms that are inhabiting the water column above the 3,000 plus meters we were looking at earlier today.
And in this area of the U.S. continental shelf, it's actually one of the widest along the U.S. eastern seaboard. So up as you get closer to North Carolina and Norfolk and Virginia area, it's a lot skinnier. And then it kind of comes back out as you go northeast around New England area. But in the southern half of the United States, it's a big, wide continental shelf. So as you said, we had to go fairly far offshore in order to get to that deep water. And currently right now, we're working in an area called the Blake Ridge, which extends from the Blake Plateau, which is where we're going to be spending a lot of our expedition this year.
Yeah, so as Casey was saying, if you're in different locations, even within the U.S., you might not have to travel quite so far off the coast to get to deep water, but here in particular, we have to travel one of the furthest distances to be able to dive as deep as we have been here.
When we were working off of Hawaii, it was just a couple hours transit to get to the same water depth. Whereas for this cruise, we had to transit pretty much a full day to get all the way out here.
Yeah, the same is true from where I did.
Here at a quick request.
Yeah.
On the stream 3, can we, instead of the quad screen, just put the echo sounder data so we can see it better? Awesome. Thank you.
Yeah, no problem.
Thanks.
Yeah, as Casey was saying, we're
Right,
pretty far offshore here, but in Hawaii, they didn't have to go very far to get to water this deep. The same is true of where I've done a lot of my midwater research.
Feel pretty good, Josh?
Submarine Canyon, just off Central California.
We could try them out.
We just had to steam out about an hour or two to get down to the same deep depths that we were at this morning. So we just reached 600 meters, so we're going to be setting up for our
The thing is, it's
our
lighting
600
up
meter,
everything
10-minute
really
transect.
Yeah, it's lighting up only the close stuff, so
So we have
minimal
some
focus.
radiolarians or
It's good.
4
It's
amp,
good if we have
radiolarians
subject
there.
down below.
Correct me
All
if
right,
I'm
pushing
wrong
out.
on that.
Get in the position.
This looks a little friendlier already.
Tina 4 off on the right.
Good job, Nav.
All right. Ready when you are, Nav.
All right.
I still haven't reached my depth.
This marks the beginning of the 600 meter depth transect, time 2050 UTC.
So we just started our transect at 2050 UTC. So I think you guys heard Nav saying that, just repeating in case that's not true. So you see, actually, see that small
You got two perfect.
animal
now there's
going
no
in
current.
circles or spirals. Now it's about top center. That
you
is
might wanna
actually
slow
a
the
cutic
ship
nav,
by 0.1.
which is an entire
Bridge, this is
phylum
RV
to
nav.
itself, or they're commonly called
Yeah, you
arrow
can go ahead and
worms.
slow the ship, decrease
And
the
they
speed
have
of
that
that,
very
move back to 0.3,
distinctive
please.
swim. Not quite sure the best way to describe it, but they're
Looks like
they're
you
pretty
got your
easy
camera
to
tilted
pick
down
apart.
pretty far.
Casey's
Camera
doing a dance
tilt
right
is
now.
at
If only you could see
Even.
it. They
It says -3.
are gelatinous worms, but then they have their head region has these retractable hooks that, when you look at them under a microscope, are rather gnarly.
Dark down at the bottom. Did you guys reconfigure
And
your lights?
their favorite
No, we
food,
didn't change
we can
anything.
actually see one of their favorite
this
prey items
guy
right
right here.
now, a copepod we're zooming in on. So those arrow worms, that's one of their favorite things to eat. And that's partly because copepods are very, very common
Coming
in
out.
the open ocean. But they're so small, you we likely overlook a lot of them. So it was great that we
Get the swimmer.
happened to see that one, so we could talk about it.
Where's he at?
Right in center. This dot.
Yeah, it's a little, looks like an amphipod actually.
Yeah,
So a
coming
type
up.
of crustacean. We don't see those a lot on their own in the water column. That one is probably out swimming around looking for a medusa to attach itself to. Commonly, if we zoom in on some of the gelatinous organisms out here in the water column, they have some stowaways, commonly have amphipods on there. And often there are very specific species that species pair. So a particular type of medusa will have a particular species of amphipod on it, which is kind of cool. So that kind of relationship between those two organisms, I think for the most part they're thought to be neutral or negative for the host, if you will, the medusa or gelatinous organism, because oftentimes we think that the amphipod is feeding on its host. So certainly the amphipod is getting a benefit out of that.
I think it's Mr. Lister.
Sometimes we think that the amphipod might be taking food out of the gut of the jelly, but I think in most cases that are known, the amphipod is feeding on the jelly itself.
Keep check. Down here better, probably? I'll breathe. Maybe I was breathing out of my mouth or something. It's a little warm in here, so sometimes.
As I said earlier, that amphipod that we saw that was just on its own might have been looking for a host. How they find a jelly out here in the dark, that is a great question that I would love to know the answer to.
This guy right here, right-headed.
That actually looks like mucus.
Come on out.
Yeah, we can keep going.
This good speed, Josh?
Yeah.
Copy. I do anticipate the current to increase magnitude as we rise up. You're at 600 right now.
Pushing ahead.
So we're almost halfway
Get
through
this guy
our.
in the center.
Oh yeah.
Slow him down.
So that is a siphonophore. That's unfortunately self-destructing. Some of these animals are so fragile that when we come upon them, they actually start falling apart.
Double dots.
I gotta turn around.
And sometimes they can do that in high currents as well, correct?
Yeah, so just the ROV replicating some high currents, and I don't know, they could probably regenerate some of that. I'm not totally sure about siphonophores, but many marine organisms can regenerate, including actually some of the squids can lose their arms as a defense or just in a predatory interaction, and they can regrow them. So it's not necessarily a bad thing that the siphonophore lost some of its feeding tentacles.
Something center.
Yeah, can we check that out? So that is a lobate ctenophore and a cyclothone fish that's swimming by right in front of it. And it's gone.
Is that right in front of me?
No, it's down below.
Actually, this might be an animal we call
maxed out.
Sedusa, which is a ctenophore that is similar morphologically to a medusa, which is a totally different phylum from ctenophores. But I'm not sure on that, actually. This is a larvation.
The big one.
As you can see, it's got this outer house that's made of mucus, and it's got, we talked earlier about marine snow, so all these particles that we see in the water column actually do still contain nutrients, so many animals take advantage of that. This particular animal, which is the small,
65.
slightly more opaque organism in the center there, and you can see its tail flapping, that produces a feeding current, collects those particles and eats them, and then discards the house when it's done.
How quickly does that happen?
That's a great question. We've actually sat around and watched them produce their houses. I think, depending on the species, they can do multiple per day or just maybe one a day. I think it probably depends on how much particulate material is in the water column, and therefore how quickly those houses get clogged up. Because they're actually not interested in those larger pieces, so they're excluding them with that outer mucus house. And actually earlier, I think it was during the 700 meter transect, or maybe between the 800 and 700, we saw a sinker, which is actually one of the discarded outer filter houses of a larvation. And because of the mucus sticking those particles together, those sinkers, as the name suggests, they sink more quickly than the particles do on their own. And that is a more rapid transportation of potentially nutritious material to the deep sea floor. We're out here we're at 600 meters right now, doing a horizontal transect through the water column. And the reason we're interested in doing this is because the water column, or the midwater, so all of the volume between the surface of the ocean down to the sea floor, is a huge, huge habitat. So even though in some cases it looks pretty sparse, so that actually looks like discarded mucus house from a larvation, or part of one.
Right, coming up.
So it looks pretty sparse here, other than the marine snow. But if you think about just the overall size of the ocean, there is a lot of biomass contained within the water column.
This guy right here.
Yeah, so that's looks like a krill.
Very close to you.
Big double-lobed eyes. Again, we were talking about, we've seen quite a few different medusae and ctenophores, and they commonly capture these krill and other crustaceans that are in the water column.
That's it.
All right, so we're done with our 600 meter transect. 2100 UTC.
Main's coming off.
And now we're gonna move up to 500 meters, correct, Navigator?
Yes, 500.
Great, thank you.
Okay.
Ready to
So we've done a 800 meter, 700, 600,
okay.
and now we're moving 100 meters shallower to do another 10-minute transect at 500 meters.
This keeps changing. It's like someone else is trying to use this computer.
Like,
he's troubleshooting it out there.
So we're seeing some more radiolarians.
500. About the same, maybe slightly larger magnitude.
It's fascinating that some of these single-celled organisms like radiolarians are larger than
leave it here and then increase it if we need
krill.
to. That's what I was, Yep.
Lighting on that last transect
Kind of a cool
seemed different. Seemed like we had less thin slip.
There wasn't as much stuff
Are there
in the water.
advantages to that?
There's a
Bridge, this is ROV now.
We didn't move anything.
That's a great question.
How much do you have left in that
You mean to being a large single cell? Well, yes,
go ahead
certainly.
And tack on another 200 meters.
One of the
thanks.
One of the features of the water column that animals need to contend with is the fact that they want to stay in the water column instead of sinking down to the bottom. So in the case of that radiolarian, we've seen a number of them. They have these appendages that stick out